Next up on the lineup, we have Life Biosciences and Jerry McLaughlin.
Good morning, everyone. Happy to be here and provide an update on our company, Life Biosciences. And I guess in the spectrum, we're probably more like a traditional biotech, and we've evolved to that over the last several years with a focus on multiple platforms, but under the housing of one company with one management team. And we continue to prosecute and execute against our business plan of not only moving the science forward, but simplifying corporate structure, simplifying our approach, really preparing ourselves for a more traditional investor pathway and traditional path forward as a biotech. I apologize there. Just excuse 1 second. And excuse me, I apologize 1 second, folks. We're having a little bit of an issue here. Are we back on?
We've got the starter slide or the title slide.
There we go. Important legal notice. So
Yes.
One thing here about Life Biosciences, it's really our belief that, as everyone has been talking about, not lifespan, but Healthspan. And to take this to a practical level, one of the areas that we really focus on is ophthalmology and visual impairment. And how we like to think about this is, what does this mean? What does Healthspan mean for the everyday patient? And when you think about visual impairment, its impact on many of us like to read, and as we age, that becomes a real problem, a real challenge for us. So we stop reading as much, and therefore intellectual curiosity is diminished. Our driving is compromised. Our ability to engage in daily activities, crossing the street, walking to shopping, walking to see friends, gets diminished. And what this ends up doing, and it's well documented in the literature, is in the case of visual impairment, it really leads to isolation. And so it gets back to this point that it's not the life in our years, but the years of our life. And we really want to extend healthy lifespan for all. I actually think a good measure is, if any of us with aged parents who are suffering from ailments, really as time moves on, what you notice is the percentage of the time on the phone that's spent talking about their diseases, their ailments, their doctor's appointments, is really a good measure of the diminishing health span. And it's our goal to have those conversations be more about vacations and trips and spending time with the grandkids than it is about their doctor's appointments and ailments. Our approach, we have a multi-platform approach, and what we're looking to do is really target the biology of aging and treat diseases where there's significant unmet medical need. And within each of these platforms, we believe we can build a diverse pipeline as well. One is Chaperone-mediated autophagy platform. We'll talk about this. It's restoring proteostasis. It's a significant issue that is just unfortunately accompanied with age. Our CMA, our ability to recycle unwanted proteins diminishes as we age, and this has effects all over the body. The second is, as we age, we have epigenetic drift, and what we're looking to do here is to restore cellular function to that of a youthful state, and this has applications across a number of disease states. And then finally, we're looking to recharge and increase metabolic burn and increase metabolism through our mitochondrial uncoupling program. And so we're targeting a number of diseases, a variety of diseases that span ophthalmology, neurodegeneration, as well as metabolic disorders. We have an experienced team where we have more of a traditional biotech background. I think the average experience level in the company is 20+ years. And really the story here is the science. The science is the story. We have science that's emerged out of Harvard, out of Ana Maria Cuervo's lab at Albert Einstein, out of folks at Virginia Tech. And we've really integrated this science into our business model. And while we maintain strong relationships, we sort of morph this into operation here on a commercial scale to move the company forward. We did raise a Series C of about $80 million. We closed that late last year. We have cash that will run our programs through the early part, if not into the second quarter of 2024. And so as we look about our business plan to support these programs, we will be initiating a capital raise, another private round, sometime in the early to mid 2023. So some of the science that was recognized in some of the major papers for each of our platforms, both in Cell, Nature Communications, and then the big Nature paper of David Sinclair's work out of Harvard, showing the impact on vision, was really tremendous. And I have to say that this has really drawn a lot of attention to the company, and we can talk about this either during the discussion here during Q&A. There's been a lot of interest in these programs from big pharma. It really depends upon the platform of who is interested and for what reason. But suffice to say, there's a tremendous amount of interest, albeit early. And so that is, will over the longer term, will be an important part of our business strategy. Looking at our chaperone-mediated autophagy platform, this is a new class of small molecule CMA activators, and with the opportunity to improve the function of multiple aging systems. So what does chaperone-mediated autophagy do? It removes unwanted soluble proteins. And unfortunately, as we age, the ability to do this is declined because of the reduced expression of the CMA receptor LAMP2A. When this happens, we have an accumulation and an aggregation of unwanted proteins, and they get supersaturated, they form aggregates, and that disrupts cellular function in the CNS, in the eye, and in different parts of the body. And our goal here is, with our program, is to upregulate LAMP2A to restore proteostasis and allow these unwanted proteins to be escorted into the lysosome and recycled into cellular nutrients as they do when we're young. We'll touch on some of the data here that we have that's been published in an Alzheimer's model and a frontotemporal dementia model. But we also have data in ophthalmic diseases as well that is not yet published, and we're pretty excited about that going forward. So once again, just looking on a very basic level how this works, we have these chaperones. They target proteins with a certain motif, this KFER motif, and they're recognized by the chaperone protein HSC70. They then are attached, and they're escorted to the lysosome, where they enter through the LAMP2A receptor. And this LAMP2A levels control this ability to recycle these proteins. These recycled cellular nutrients are then recycled throughout the body and reused. And if you look on the right, unfortunately, both in mice and in humans, LAMP2A levels significantly decline as we age. You can see the difference in the humans from a 25-year-old to a 65-year-old. And herein lies the problem. What our CM activators do, they upregulate this LAMP2A and allow us to continue to recycle these unwanted proteins. So looking at some really interesting data here of our tool CMA activator compound in a frontotemporal dementia mouse model. On the left, you see in a control mouse at 9 months, you have no accumulation of mutant tau. In a PS19 mouse, it's mutant tau model, you see significant accumulation of tau in the center. And then after introduction, over the course of 6 months with our CMA activator compound, you not only see a lack of progression or accumulation of tau, but you see a removal of tau in these 3 key areas of the brain. So that's check 1, able to remove tau. And then looking to the right, the question was, could our CM activators not only remove the tau, but could they impact behavior and memory? And in this, on the right, you see an open field test. The control mouse, mice know to stay on the outside of a field, right? For safety reasons, it's programmed into their DNA. What you see with the mice, the PS19 mice with significant tau mutations, they forget and they cross the dangerous areas of the open field, introduce our tool compound, and you see a restoration of this ability and this behavior to stay in the outer sides of the field. Then looking at memory about a mouse memory model, you see in the control mice, and then you see in this PSC19 mice a reduction in memory, and then a restoration of this memory back to that of a control mouse. So we're really excited about this data. We're also seeing some really interesting finding, and I mentioned in some ophthalmic indications, and we're excited to move this program forward. This is a novel approach. It's an entirely new target. We're out in front. The work by Anna Maria Cuervo at Albert Einstein is groundbreaking. It has drawn a lot of interest across the industry, across the pharma industry. We continue to have those conversations. I will say we would prefer, and we will continue to advance the programs, and as we are in a preclinical stage. As I mentioned, we demonstrated benefit both in ophthalmic and neurodegenerative preclinical studies. We have a lead for an ophthalmic indication, and we expect to initiate IND-enabling studies in the early part of 2023. Turning to our gene therapy, our epigenetic reprogramming platform, the whole goal here is to restore cellular function to that of a youthful state. As mentioned earlier, the epigenome consists of chemical modifications of DNA and proteins that regulate this gene expression. Unfortunately, common theme here, as we age, we have epigenetic drift, and this really leads to disrupted, dysregulated gene expression. And our whole goal of this program is, and the hypothesis was, could we restore the epigenome and restore cells to a youthful state without bringing them all the way back to an embryonic state where they lose their identity. We'll talk about that in a second. So at the top level, we have demonstrated proof of concept in glaucoma models. We have a optic nerve crush model. And importantly, with our gene therapy, we've had no adverse safety findings, no teratomas in mice after 15 months of delivery. And the reason behind that, we believe, is that we all know about 4 Yamanaka factors leads to full reprogramming. There's a Nobel Prize. These epigenetically younger cells unfortunately move back to an embryonic state. They're embryonic-like stem cells, and they lose their cell identity, and they quickly form teratomas. The novel invention here was looking at partial reprogramming, removing c-Myc, and so having 3 Yamanaka factors. And what was demonstrated by David Sinclair at Harvard was that we could actually bring these cells back to a youthful state, a younger state, and retain their original identity. And as I mentioned earlier, importantly, we've been able to show safety out to 15 months with this partial reprogramming. So a quick look at some of the data. We know that when injured or untreated adult optic nerves, these retinal ganglion cells, they don't regenerate. Just doesn't happen in the human body or in animal models as well. What this test here was to look at an optic nerve crush model, taking 1-year-old mice and crushing the optic nerve, and then either inducing sort of a control gene therapy or our partial reprogramming gene therapy. And what you see highlighted here is our OSK gene therapy had significant new nerve growth past the crush site. This was 5 weeks post-crush, 5 weeks post-crush. So for the first time, we're able to see this nerve regrowth. And the question there is, that's great, but is this nerve growth without activity? The next model was in a mouse glaucoma model. We were able to demonstrate restoration of electrical activity and vision after vision had been lost in this mouse glaucoma model. You can induce glaucoma in this mouse model, and what you see in these mice with glaucoma, you see a significantly reduced amplitude or electrical activity in the eye. And then when you introduce OSK, our gene therapy, you see a restoration of electrical activity. You then can look on the right here and see a restoration of visual acuity. In the dark gray, you have normal mice, you have the visual acuity with these mice with glaucoma in the green, and then you see after 4, 8, and 12 weeks of treatment, you see increased visual acuity in those treated with OSK. So we're real excited about this data and its ability to not only in a disease such as glaucoma, not only slow the progression, which is all we've been doing with our therapeutics over the last 50, 60 years, but for the first time, we hope to be able to restore vision and restore some level of vision for these patients and reverse the disease process, at least for some period of time. So where are we? We have intravitreal injection demonstrating this proof of concept. As I mentioned, the safety is very important here in gene therapy. We have an ongoing non-human primate study and an ophthalmic indication, and we expect to have that readout by year end. That's a very important and very exciting study for us, and we look forward to sharing the results of that study once we have them. Finally, our mitochondrial uncoupling platform. New class of, this is once again a small molecule for treating metabolic disorders. We all know about DNP, and we all know that mitochondrial uncouplers are known to extend lifespan in preclinical species. And an old molecule DNP from the 1920s, patients were losing on order of 2 to 3 pounds per week, but it proved to be toxic. Things like hyperthermia, among other serious adverse events, result in the removal. And there's been a long history and a hope that someday we can solve the safety issue with DNP. And what we believe is that we have a new class of mitochondrial couplers that can enhance metabolism and result in reduction in fatty liver and liver inflammation in NASH, but also weight loss by mitigating the risks, the safety risks of DNP. We believe they're self-limited in nature and can address these toxic effects. And once again, how this works, the mechanism of action, there are pumping of protons into the intermembrane space. This takes caloric burn for this to happen. The protons pumped into this intermembrane space release energy required for ATP synthase. And this happens when they move down their concentration gradient through a channel called ATP synthase. What our mitochondrial couplers do, they allow protons to leak out of the intermembrane space independent of ATP synthase. And what this does is it forces more caloric burn for an equivalent amount of ATP synthase or energy production. So by that mechanism, we're able to increase the metabolic rate. And the real interest here is that if you look at the obesity space, almost exclusively the mechanisms are all about caloric restriction, right? Reduction in intake. And here you have with mitochondrial couplers is the ability to enhance caloric burn. So not only as a monotherapy, potentially as a combination therapy, that's the real interest here for mitochondrial couplers across the variety of metabolic disorders. Another key finding in our data is that, and this is what you want to do here in the red, you see mice with the Western diet, and when you introduce one of our tool compounds, you see a significant reduction in fat mass, and on the right, without compromising lean muscle mass. And that's really important here, whether it's for NASH or obesity. And so we're excited about that we're able to do that, reduce fat mass while maintaining lean muscle mass. So where are we with this program? We have preclinical studies that show both weight loss and reduction in fatty liver and liver inflammation. We're in the process of developing a lead candidate with the desired product profile. In this class of agents, it's really all about safety, right? We know that mitochondrial couplers are effective, and then it's really about overcoming that history and with DNP and developing safe mitochondrial couplers for these metabolic disorders. So that's our story in a nutshell. And once again, we are well financed through the first part of 2024. We intend to kick off a financing next year. Our current monies will take us up to the clinic, but not into the clinic necessarily. And we'll be looking for that next round of financing to take us through phase 1 and potentially into phase 2A studies. So with that, I thank you, and I'll open it up for questions.
Thank you, Jerry. Phil, Dennis, do you want to go first?
Yeah, great. Hey, Jerry, how you doing?
Doing great.
So, Jerry, obviously the work that you're doing is platform-based, which is what a lot of people are looking for at the moment. But of course, 3 very diverse portfolio assets that you're working with. Is that difficult to manage in terms of both your research and development as well as your IP protection?
Yeah, I'd say, let me take in reverse. In the IP protection, it's really just about resources, right? And investing the capital. And yeah, so that's really the only issue there. I'd say from a management perspective, we have built up a system. We maintained a very lean operation. We have 10 FTEs. We have a number of collaborations with CROs. I think it is a challenge, Phil, and I think it's not tenable over the long term. But I think at this stage we can manage when we're in the preclinical stage. I think it's what it's going to force us, and I think it's what our strategy, and I mentioned this very early on, is that depending upon the platform, depending upon the disease state, there's different interest levels among pharma in each of these platforms. And I think we'll have to be proactive in terms of looking to partner one or more of these programs sooner rather than later, because it'll become untenable, I think, from a financing perspective and from an organizational perspective to run these platforms over the long term.
Mm-hmm.
So I think that's probably the biggest thing. At this point, though, I think we have the resources, we have the expertise to prosecute, I think, through IND enabling studies.
Yeah, I mean, a lot of encouraging data that you've got there. I mean, it's fascinating to see what you guys have been up to.
No, great. It is exciting. I think one of the things that Phil, I think it's worth mentioning in our approach is we are taking the classic biotech pharma approach of rigor, and having been on the other side in big pharma, we are employing that with each of our programs, and I think what that's going to allow us to do, and this is the benefit of having multiple platforms, we can be highly discerning, right? We are going to have the approach that, when we're in our medicinal chemistry, a quick kill is as good as holding on to a molecule, right? We're going to learn fast, make quick decisions, and with 3 platforms, we can do that with a discerning eye, right? We'd love for all 3 of these platforms to work, but we know the risk of our business, and we won't be afraid to make those tough decisions as we go forward.
Yeah, great. Thanks, Jerry.
May I interrupt with an interesting question that seems to be coming up more and more? Are you able to parse out the data in your mice between male and female mice, especially as it relates to metabolic rate?
I don't believe we looked at that. It's an interesting question. It's something we'll bring back to the team, but we have not looked at that yet.
Okay. And I'm also curious, when I look at your leadership, I don't see females, and I'm curious if there is a strategic plan to add some to the roster.
Yeah, I think that, so we had, and we had some departures earlier in the year, and of course, yes. And if you look at our organization overall, it is balanced in that fashion, but it is of importance to us going forward, both at a management level and a board level. Diversity of thought, diversity of gender, diversity overall is very important.
Wonderful. And one last science question. From the standpoint of what's challenging in the diabetic retinopathy drug therapy world, is there a potential opportunity to move from an intravitreal injection to some other way to administer this ophtho drug?
Yeah, so we have 2 approaches with our Chaperone-mediated autophagy program. We are so for that program, which there is ophthalmic implications, we are starting with an intravitreal injection for an ophthalmic indication, but we also have a systemic program looking at neurodegeneration. And our goal from a life cycle perspective would be to explore small molecule or oral administration for ophthalmic conditions as well.
Sure, that's great news. Wonderful. How about you, Dennis?
Hey, Jerry, it's Dennis.
Hey, Dennis.
Great science, man. It's amazing. Congrats. The whole gene therapy field, how do you see the risks to your lead program? What are you worried about?
Yeah, so I think there's safety, right, is one of the big questions. And I think emerging is manufacturing as well, right? I think one of the things we're spending a lot of time, Dennis, the next step for us is to kick off a GLP manufacturing campaign, which is not for the faint of heart, right? It's an expensive proposition. And we've spent an inordinate amount of time evaluating the vendors. We've brought in expertise from folks, like at Spark, who've run their programs, to ensure that we're not only on top of the technology as it relates to our delivery and our capsids, our vectors, but also in terms of the the contract manufacturers in the field and their expertise, right? Because they're popping up, and to make sure we're picking the right partner, and then also keeping a keen eye on the regulatory environment. Because what we're seeing is as there's emergence of expected AAV, we're going to be an AAV too, right? So as there's going to be a preponderance of these programs moving into the clinic, the agency now is becoming more discerning on specs, and so we're trying to stay ahead of that. So I think it's a lot of we're sort of measuring. There's the old saying, measure twice, cut once. We're measuring 3 times.
Yeah.
Because it's an evolving field. So we're keep monitoring safety constantly, and we feel good about where we are for what we have now, right? As best as we can. And then on the manufacturing front is really being discerning and careful in our planning for manufacturing.
A stunning phase 3 or Series C amount of money. Who are your kind of 3 or 4 biggest shareholders?
Yeah, so it's a number of undisclosed family offices, but then the lead investor on the Series C is Alpha Wave Global. And Dennis, what's interesting, I think what we're seeing is someone like Alpha Wave. They have obviously a good name in investing, and they've been building up their biotech, institutional knowledge and expertise. So it's while they're not traditional, they're not ACE-ling, right? We see a number of players who are sort of leaning in a little bit into our space.
Yep.
And so as we go forward, I think you've heard this earlier, a good desire here would be to bring traditional biotech investors. I think our story, given its more traditional biology, disease, unmet need, novel science, that said, obviously we have to create optionality and be prepared for any investment pathway.
Well, congrats on the progress so far. Great. It's great.
Thanks, Dennis.
Dennis and Jerry, how common or rare is it that when you have a large round, you keep investors confidential? Jerry, I'm not putting any pressure on you. I'm just asking about the phenomenon, because usually you have a sizable round.
Yeah, it's not unusual.
Okay.
It's not unusual. It's up to the company.
Yeah.
No problem.
Yeah, so we share Alpha Wave because they're on the press release and they're willing, they were happy with being out there in public. Yeah, but it's very common. And typically when the next round, because when you're under CDA, Thomas, then obviously there's a disclosure under CDA of the cap table.
Sure.
So Jerry, given what you've laid out in the planning, provided that obviously you've got the funding, you definitely have the support of science and a great plan. And for those of us in this small group of peers for longevity related companies, we're all looking at you to sort of lead. You're a year away from clinic, or roughly a year away from clinic?
Roughly a year away from clinic, all pending our IND enabling studies, right? We have a couple flips of the card, and if they meet our target product profile and data is successful, we'll be able to be in the clinic with one of our platforms. And if not, it'll be the following year. But we sort of have a runway set up. Each of our platforms is about a year apart from entering the clinic.
Fantastic. Well, we will watch with great anticipation. And thank you so much for a great presentation and your time today. Best of luck, Jerry.
Thank you for the opportunity. I appreciate it.
We're rooting for you, Jerry.
Thanks, Dennis.
Thank you, Jerry.
Yeah. Wonderful.